don't look at. But we thank God for the HIV DNA testing that actually made this popular, and some other malaria tests that actually make DBS testing popular. And of course, I'm also quite interested in the organism that will be used in this workshop, which is something that I hope we'll learn a lot from. So, like I said, it is simple, easy collection. Now you can use it to collect transport samples. Maybe you want to do DNA extraction, RNA extraction, ready for processing in minutes, efficient, reliable, fast. Storage is no big deal. You don't have to buy very big -80, for you to store your samples for a long time. But of course, everything always looks perfect when it comes to us. Before we begin to see the other sides of what we have. Next slide. So like I said, these are a lot of tests have been done using DBS. This is just one slide out of several slides that we got this information from, and we can see a lot of viruses. I'm sorry, my boss is a virologist, so I'll be talking from the point of viruses. A lot of viruses that they've actually used it for. Next slide. So what is a dried blood spot? This explains it all. It's just a filter paper where you can actually get a small speck or drop of blood, and you just put it in those circles like they've explained, and it's used routinely like all the things I've explained. Just put it there, you label normally the name, and you cover, and it sounds that easy, but it has very specific procedures that we have to follow, which we'll find in the subsequent slides. Next slide, please. So what it requires, let's compare DBS and plasma, for example. Yes, venipuncture, yes, for plasma, because you need to get quite a number of volume. It doesn't require centrifugation, so you don't have to bother yourself about centrifugation. Stable at room temperature, yes. Biohazard for shipping purposes. For plasma, yes, there is a lot of biohazard because you need to follow the IATA regulations for transporting some of this specimen. And unfortunately, in some parts of Africa, we don't follow most of these regulations. We just collect the blood sample, put it in, some will even wrap it in paper, in the request form, and they can transport it from Lagos to Kaduna. And we think we are transporting samples. So at times, when samples come to the lab, at times, probably it's leaking, we have the specimen on the side, and if you're dealing with some very dangerous pathogens, you have to be very careful. So if you have strict policies, at times you may have to reject some samples. I'm sorry, although you won't give them back the sample to go away with, keep the sample, we are not going to process the sample. So you get another sample, package it properly, and send to us. So you require dry ice for shipping. Dry ice is expensive, But things we need to consider when of DNA from white blood cells, right? Because you're spotting the whole blood. At times you want to do viral assays. You want to monitor viral loads. You want to look at it in the serum, rather than the whole blood, because the whole blood can contaminate the assay, and you can begin to have unspecific amplifications that can give you false positive results. So most of the time you try to avoid that, but in DBS it's a bit difficult, except you spot the plasma. And for some assays they frown at spotting plasma. The risk of molecular contamination, big problem. You can get molecular contamination. Take for instance, if you don't dry your paper very well after spotting, fungi can grow on it, bacteria can grow on it, all sorts of other microbes can grow on it. So in the end, we have molecular contamination. All of biohazard risks, because you need to puncture where you spotted, right? So you must use punctures that are sterile. And for laziness at times, if the lab supervisor is not watching properly or monitoring closely, you can have cases where somebody will just use the same puncture. Or guy is not here. Just do it. I can't sterilize this overnight, or I can't keep myself. And before you know, the person has transferred sample from one person to another person, and if you're doing sequencing in the end, you'll be seeing some funny results and you'll be wondering, last week this person had this genotype. This week this person is having another genotype, and you begin to wonder where the problem came from. It's from, when we get tired in the lab at times and you want to rush the whole thing, and we do that. So viral load measurement from DBS, is it equivalent to plasma? I don't know. This would require plenty of optimization. It could be, because as a scientist, I don't believe in no. It could be. You can optimize if your lab has the capacity to develop and optimize your protocols. You can optimize your protocol in such a way that it could be. But if you are using ready-made kits that they ask you mix A plus B plus C plus D, you have to wait for the manufacturer to give you the guidelines on how to go about it. Next slide, please. So, what materials do you require? So, there are different types of papers, right? Different types. Some people even take ordinary filter paper in the lab, but I don't subscribe to that. Let me just spot ordinary filter paper in the lab, because most of these papers are actually impregnated with some reagents that are karyotropic. So if you put anything on it, they denature anything that comes in contact with it. But if you just put your Waxman paper in the lab and you spot it, did you impregnate it with any karyotropic agent? No. So you can have a lot of problems with that. Even the karyotropic agent is actually supposed to even preserve your DNA. Because once you use it to preserve your DNA, to preserve the RNA, preserve your protein. But on the ordinary Waxman paper, you have to rely on your storage. Your storage, your drying, and your storage has to be perfect. If you have issues with humidity, you will always have issues. So I don't subscribe to and transport the sample. So always use commercially made ones. And commercially made ones, this is one of them. It comes with a paper where you can spot, you have up to like 1, 2, 3, 4, 5 holes. It comes with a desiccant that can help you absorb in case of humidity, so that fungi or some other microbes will not grow. It comes with an indicator that can, help you tell the humidity, whether you should change the desiccant or not. Some would even come with, what do you use in pricking?
Lancets.
Lancets, yes. Lancets that you're going to. Some will even come with swabs, alcohol swabs that you can use. So it's always nice for you to have this. And once you have this, next slide, please. So how do we collect and dry DBS? So clearly label the card with identification number. Of course, we are all scientists. We agree that labeling is extremely important. We've had a situation where, somebody mislabeled a sample and it became a WHO issue. Just somebody. The least person that we expected would miss it. Somebody that collected the sample labeled A for B, and you said A had this disease, and when they got to the place to investigate, the A had everything opposite to what the lab said. So lab, what happened? Did you label wrongly? Okay, where are the samples? Run the samples in front of me. Start from A to, okay, still came out the same way. Okay, send it for sequencing. So who actually owns the sample? Until one day they now discovered that the person collecting on the field will just label anyhow. And just as it's collecting, it mixes everything up and sends to the lab. So labeling is extremely important. Then your pipetto. Like I said, the DBS card can take between 250 and 500 microliters. But you don't want to, spot in such a way that you come outside this circle. So it's recommended that you use like 50 to 75 microliters. So that once you put it, and don't try to repeat spotting. Somebody, at times in this part of the world, you want it to be strong. You want to repeat something, so you keep spotting and spotting. So don't try to do that, just spot at once, and you leave it. But you will also observe that the pipetto has a barrier filter. Can we see it clearly? There's a barrier filter there. And if you're doing such works, don't try to use barrier filters. The reason is that you can have cross-contamination. While pipetting, if the spring is not, of course, we expect that your pipetto should be calibrated. It could jump up you know, and because you're doing DNA work, the slightest contaminant would cause an effect. So use a barrier filter. So whatever happens, this barrier filter can pick it up. And if you're working with very dangerous pathogens, I strongly recommend use a barrier filter for what you're doing. Except even if you are doing research, just try and use barrier filter for the purpose of this period. Next slide, please. At times, some people want to spot from when you've collected. Next slide, please. Aha. See, collection from finger prick. You know, just you. Some recommendations would advise you prick from either this side or this side, and don't try to milk it too much. You know, some people try to milk and milk and milk. Just get a drop. A drop is approximately like 20 or slightly above 20 microliters. Just put it on top. Once it's there, it's perfect. It would expand, and don't try to put and put and put and put. So, let it uniformly saturate the entire sample. But of course, you have to follow the universal safety precautions. Wear gloves, wear your lab coat. For me, I don't take chances. In fact, at times when Ebola came, I began to run away from bleeding. So when people are doing how to bleed, you know, because you never can tell. You know, you see all sort of cases and you're scared. So next slide, please. So do not touch the areas of the card that will be used. Don't ever use your plain hands to pick the cards. Why am I saying this? Because your hands can transfer DNA materials to the cards. And that would also cause some effect. So don't ever. Just pick the card with your gloves and try not to contaminate it. Then don't apply blood to both sides of the filter paper. Like I said, you want it to be well emphasized. You put the other side, then you put it, and they will get enough to use. You know, it's okay. Do not apply blood from more than one patient on the same collection card. You know, at times you want to manage. Please don't try that. Just use one card. You know, God will provide. Research grants will come, and government can subsidize. So do not press the filter paper against the puncture site. You know, when you are collecting, you want to press, and so that things. Then do not milk the finger. You know, some of us would have. I want plenty blood to come out, and you milk. Next slide. So this is one of the most important parts that scares me a lot about DBS. You need to be well proficient in handling this aspect. You have to be able to dry your papers. There are racks, there are paper racks that are being supplied even by the suppliers of these papers that you can use to dry horizontally. Don't stack the papers on top of one another while drying. Dry at room temperature. Some people try to do this in the biosafety cabinet. Just put it in your biosafety cabinet. Just leave it maybe for some time. Then do not touch or smear spot. If you're going to do it in your biosafety cabinet, please make sure your cabinet is on. Don't put it in your cabinet and you now put on your cabinet. You know why? At that point, when you put on your cabinet, it draws air from outside. So in trying to purge the air inside, there is this circulation of air that would mix with your sample, and before you know, you can have issues. So put on the biosafety cabinet, let it purge for a while, then you can work in the biosafety cabinet. Then do not touch or smear spots. Do not dry in direct sunlight. You know, like common sense would tell. Why don't I just dry it in sunlight so that it can dry faster? Then do not heat, stack, or allow DBS to touch each other. So that's next slide. I'm sure all of us would prefer to dry like this. You know, nobody would prefer to dry in this way. You know, obviously you've transferred a lot of particles from the bamboo, and a lot of microbes would have been transferred. Next slide, please. So when you're done, you insert into a sealable plastic bag, which comes with the kit most of the time. Add your desiccant packets, and we all know why. Add your humidity cards, if you have humidity cards. Then label contents of the bag and seal. Next slide, please. So we see, I see a lot of this in AP lab. Next slide. So your desiccant goes in. Next slide. The indicator. If it turns pink, to tell you replace. Next slide. So you're done. Label. That's after you've dried, not before. After you've dried, this is what happens. Next slide. So you insert your bundle DBS into resistant envelope, include appropriate documentation. Then next slide. So these are some of the examples of issues that we may likely have. This image is not showing clearly here like it's supposed to show. At times we need to be careful. You have to be ensured that it dries. So just leave it, don't be in a hurry. Just ensure that it dries. Then at times when you have serum, when you are milking a lot, you can have serum rings coming out. At times when it's scratched, maybe you mistakenly pack them together, you can have it scratched. Then obviously this is no blood. But in biology lab, it's not only blood that you can use. At times you work with DBS, phosphate buffered saline, and you won't see anything. So if you're in the biology lab, be careful. Maybe they've spotted Ebola and it's not showing anything. But if you're using blood, definitely you should have blood. You can see more of invalid specimen. You can see quantity insufficient for testing. The same, not dry, super saturated, diluted, discolored or contaminated, and a whole lot of things like this. Next slide. So once you have your DBS, maybe you stored, now you want to perform your PCR. The next thing is you want to purify your nucleic acids from it, right? So the first thing you have to do is, you cut. I like this cutter, very nice, very lovely. You cut it out. You transfer. Some people, there are different methods of purification. So you have to look at the methods that you have in your lab. But one of the easiest methods that we use is the spin column method, which gives the cleanest nucleic acid you can ever think of. So what you now do is that you try to lyse the content, of the DBS card cut in your lysis buffer. And what is the function of the lysis buffer? What the lysis buffer does is that it breaks the cell membrane, the cell environment, and so that the cell can release its content. And once the cell releases its content, there are some other reagents that they would have added into this lysis buffer that will make it be able to, by the time you now go through, so in the end, you want to dilute our nucleic acid from here. If you have grade 1 water, HPLC grade water, just add 50 microliters here. Your very clean nucleic acid will come out. But usually we use TE buffer, choose EDTA buffer, and you get what you're looking for. Next slide. So I felt happy when I first met DBS. So I thought I could spot all microbes on DBS. But in our discussion, I discovered that there are some pathogens that the CDC calls priority pathogens. And when you look at priority pathogens, when you look at the category A, B, and C, and you look at the types of organisms that are there, like anthrax, botulism, plague, smallpox, tularemia, viral hemorrhagic fevers, and the rest like them, you have to be careful when using some of these cards. So we would not recommend that you use some of these cards for these highly pathogenic organisms. Next slide, please. So what is recommended is that you use a triple packaging system. Because if you are transporting Ebola sample or Lassa fever sample, WHO expects that you use the IATA certified method, that has a triple packaging system. You will notice that your infectious substance is in a container. The container is wrapped with an absorbent pad. That absorbent pad, you can use your hypochlorite, your hypochlorite solution to wrap it. This goes into another container. This goes into another container that's properly labeled. And this also goes into another container. You can see the level of packaging. And there are some higher levels that you want the virus, or you want it to be cold. Let's say that you want to work with large genomic sets or genomic data, or you want to amplify the whole genome of the virus. We usually recommend that you do your transportation in this way, and you add ice So there are some triple packaging systems that have ice compartments that you add to it. So DBS is good, but just take note that you can't use it for very highly pathogenic organisms, but you can use it for other types of commonly used organisms in the lab. Next slide, please. So discussion. So how do we then transport and test such categories of pathogen? Well, I'm looking forward to learning from all of us because I'm a life learner, and these are some of the technologies that people have come up with. This person came up with this, that if I spot the blood, I can close it. Somebody said in my lab that, what of drying? Is it going to dry? How will you dry it? You know, people began to come up with all sorts of things. Another person said, why don't you collect the organism? You can inactivate the organism. You know, you can use heat. Yes, heat will inactivate. You can use low pH, you know, to inactivate. One very common one that I know, you can use treating X detergent or SDS to inactivate some of these organisms, and you can now spot them on the paper carefully and have them transported. So I look forward to learning from you, like I said, and I thank you so much for listening. Any questions, please?